Usually, Professor Aartsma-Rus gives an overview, but this time, as Givinostat is quite unknown by a lot of people, it was better to give more time to one product and spend a lot of time, or a lot of different subjects having a little time on that. So that's why this was decided. So.
Okay, thank you. And you should now see my Givinostat slide.
We do.
So as I said, I will now go into my disclosures. So I am advising and consulting a lot of companies, including Italfarmaco, which is the company that produces Givinostat. And so just so that you know, when I do this, they pay the LUMC, not me personally, but just be aware that I am advising them. So for full disclosure, and there's a list of other companies that are for this topic less relevant, but just know I'm not exclusively consulting for Italfarmaco. So when we want to understand how Givinostat works, we first have to look into a little bit the pathology of Duchenne. So Duchenne patients don't make dystrophin, and dystrophin normally connects the skeleton of the muscle fiber to the connective tissue surrounding each muscle fiber. And in Duchenne patients, this connection is lost. And as we know in Becker patients, we have a connection, but the protein is shorter, so it's partially functional. So when there's no dystrophin, what happens is that during muscle contraction, there's continuous damage. And that leads to chronic inflammation, scar tissue formation, and then, so this is fibrosis and adipose tissues. Regeneration and the repair of muscle is impaired. And of course, that leads then to the chronic loss of muscle tissue and muscle function. So to understand how Givinostat works, we first have to understand a bit how normally our muscles are repaired. So when healthy individuals damage the muscle by doing stupid things like exercising too much, going to the gym, and putting in too much effort. Many different ways that you can damage your muscle. So when there's damage, if you're healthy, this is not a problem, because there's a set of processes that will correct for this. So first, the debris will be cleared by immune cells, and these cells are called macrophages. So the macrophages will clear the debris. Then the FAPs, the fibroadipogenic precursor cells, they will repair the structure. So they will make new connective tissue. So they make the scaffold of the muscle fibers. And when the scaffold is there, then the muscle stem cells are activated and they will repair the muscle. So they will fill these tubes of connective tissue with muscle, which is again the contractile muscle. So these processes are very well synchronized, because you first need to clear away the debris, then you can start repairing, and only when you have the connective tissue repaired can you repair the muscle. And like I can explain it, compare it to when you remodel your kitchen. When there's a gas explosion in your kitchen, you're gonna first clear away everything, all the crap, all the rubble. Then you will repair the structure of your kitchen, say the walls, etc. And only then will you add the content of the kitchen, like the cupboards and the appliances, etc. So it's the same with the cell, with the muscles. You clear the debris, then you repair, and then you regenerate the muscle. What happens in Duchenne patients is that there's chronic damage. So there's always damage somewhere. So this means there's chronic inflammation. And the problem is that normally in healthy individuals, the inflammatory cells, or these macrophages, will give a signal that as long as they are busy with clearing away debris, they cannot, the repair should not be started. You first need to clear away the debris. Now there's chronic damage, so there's always damage happening somewhere. So these FAPs, they start making the connective tissue, but they make too much because they get the wrong signals. And the muscle stem cells, they never get the signal to start repairing. Instead, they get signals to also become FAPs. So they differentiate from muscle stem cells into these FAP cells. So because there's chronic damage, the whole system goes haywire and the repair is inhibited. So this is how things go wrong. And then to go to the Givinostat mode of action, we have to go very deep into protein modifications. That is the histone acetylation. And why do we talk about histone acetylation? Because Givinostat is an histone deacetylase inhibitor, so an HDAC inhibitor. Why is this important? So we have acetyl groups, which are small chemical groups that can be added to a protein or can be removed. And this basically, if you add it or if you remove it, the protein will be in, say, turbo format. And if you add it, it will be in the eco type. So proteins can be very active, and then you add the acetyl group, and then they will be less active. Also for DNA, it's important because our DNA is wrapped around proteins to make sure that they're not torn, because well, we have 2 meters of DNA. If this just lies around in a small cell, it will start to get knotted, and then it can break, and that's not good. However, if you wind this DNA so tightly around proteins, you cannot get the DNA to be transcribed. So you cannot get the RNA, you cannot get the protein. So also this is modified by acetylation, because if an acetyl group is added to a histone protein, then the DNA will open up and you can get your gene transcribed. So you have enzymes that add acetyl group, these are called HATs, histone acetylases, or remove, and these are the HDACs, so the histone deacetylases. What happens in Duchenne is that the enzymes that remove acetyl, so the HDACs, are more active. And this is not good for pathology because now the DNA is more closed. So all sorts of proteins that are needed are not made when they should be needed. And this means that the inflammatory response is not dampened, but is chronically active, which is a big problem. It also means that this fibrosis of the fibs that is stimulated. So they make too much fibrotic tissue, they make too much connective tissue. And the enzymes that are involved in doing this should be, not be in the turbo mode, but because these HDACs are more active, they are in the turbo mode, which again leads to more fibrosis. And then regeneration is suppressed as well due to this. So if you see that Duchenne is worse because these HDACs are too active, then you can say, well, what if we inhibit these enzymes? That should then have a therapeutic effect. And this is what Givinostat does. So Givinostat is an inhibitor of the enzyme that removes acetyl group, so the HDAC inhibitor. So what should this then do? This should dampen the immune response, so you would have less muscle damage. The fabs are reset, so they don't get this signal from the macrophages to make too much connective tissue. This the signaling pathway that is in turbo mode is now out of turbo mode. So they do what they're supposed to do, which is supporting the muscle stem cells to repair. So repair is improved. So this is the theory, but of course what's important is what happens in reality. So in mouse models, this is indeed what happened, but what is more important is what's happened in humans. So clinical trials have been done for Givinostat. And in the first clinical trial, they treated patients for 1 year and they took a muscle biopsy at baseline, so before the treatment, and after 48 weeks, so at the end of treatment. And then they measured how much fibrosis the patients had. And all patients had less fibrosis after 1 year than at baseline. So some patients, so the extent of the reduction differed for patients, but all patients had less. So that was of course promising, but more important is what does this do to function? And then a larger study was done, placebo-controlled, in ambulant patients over 6 years of age. And there the primary endpoint was met. So patients who were treated managed to climb 4 stairs quicker than patients who were in the placebo group. And also secondary endpoints improved. And then you can think about other functional tests such as North Star, but also MRI showed a reduction of fat infiltration in the treated patients compared to placebo. So this all fits with what you would expect from this approach in theory. So the current status is that Givinostat has been FDA approved for patients 6 years and older since last year. Very recently, it's also now conditionally approved by the EMA for patients 6 years and older who are on steroids. But as part of the condition, a confirmatory placebo-controlled trial has to be done by the company. And the evaluation by the MHRA, so the UK regulators, is ongoing. So looking toward the future, so there's evidence that HDAC inhibition reduces pathology in mouse models and Duchenne patients. But of course, this is a new treatment, so we don't know the long-term effects yet because it's new. What it doesn't do is address the lack of dystrophin. So dystrophin is still not there, so the pathology will still continue. You can, of course, restore dystrophin with other therapies like exon skipping and AAV microdystrophin gene therapy. But also there, the pathology will still be ongoing because exon skipping leads to Becker-type dystrophin and microdystrophin is small, so this is not fully functional. So also here, there will still be pathology. So most likely in the future, there will be a combination where you try to address the pathology with Givinostat or other compounds that can target pathology and restoring the dystrophin. But that's, well, for the future. If you want to know more, I have again QR codes for you. So I wrote an explanation of how Givinostat works in text, and then Ital Pharmaco sponsored me to turn it into an illustrated cartoon. So you can get both of them. And like I said, this is the cartoon is sponsored by Ital Pharmaco, but it was an unrestricted grant. So I gave the input to the people who illustrated it, and they just paid the bill, but it's good to know that they did pay for this. So that is if you want to know more. So I'm assuming that people that want to know more have now scanned these QR codes. And with that, I will thank you and I will